Hello everyone. Am I audible to all?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes. Welcome participants to our engaging webinar on breast cancer research and the power of patient-derived organoids. I am Sushi Swagatika, your enthusiastic coordinator for this enriching session. Get ready to embark on the exciting journey with Heredity Bioscience. Heredity Bioscience, a renowned biotech company and research center based in Odisha and as well as Guwahati. Proudly presenting this eye-opening experience, discover the wonders of videos and their roles in understanding tumor biology. Before we proceed, I would like to introduce our esteemed speaker for today, Dr. Abhijith Kuttanamkuzhi from Princess Margaret Center of Toronto, Canada. All the forefronts of breast cancer research, Dr. Kuttanamkuzhi brings a wealth of knowledge and expertise in the field of patient-derived organoids. We are fortunate to have him share his invaluable insights and groundbreaking research with us. Get ready for an enlightening session as we develop into the cutting-edge advancements in the breast cancer research with Dr. Kuttanamkuzhi. I would like to take a moment to introduce our esteemed convener for today's webinar, Dr. Gopal Purohit. As the CEO and co-founder of Heredity Bioscience, Dr. Purohit is a visionary leader with a profound impact in the field of clinical genomics and infertility research. With a wealth of knowledge and experience, he led groundbreaking projects that changed how we understand and handle genetic factors in infertility. Dr. Purohit's unwavering dedication to advancing scientific knowledge and patients make us a pillar of our organization and a great mentor to countless researchers. Before we proceed further, I would like to share some important information about the schedule and instruction of today's session. We kindly request all participants to stay until the end of the webinar. At the conclusion, we will provide a feedback link for you to share your thoughts and suggestions. Please note that only fully completed feedback forms will be considered able to cooperate participating in discussion. We request that you maintain discipline and this will ensure a smooth now I am delighted to hand over the virtual stage to our welcome to all the participants. Let's make the most of this educational opportunity and learn together. Thank you and over to you, Dr. Purohit. Please, sir.
Is it my slide visible? Anyone?
Yes, sir. Yes.
Thank you.
Yes, sir.
Good evening, everyone.
Very good evening.
Thank you, everyone, for joining us today on this exciting journey into cancer biology research. So your presence here in the testament to your commitment to advancing knowledge and understanding in this critical field. I would like to extend a special thanks to Susri for the kind introductions and putting together this esteemed event. I am Dr. Gopal Purohit, CEO and co-founder of Heredity Biosciences. It is a pleasure to have each and every one of you here with us. It is a true of honor to be a part of this webinar and to have the opportunity to share valuable insight with all of you. The topic of today's webinar is in vitro preclinical model in breast cancer research and importance of patient-derived organoids. I would like to take this moment to express our deepest gratitude to our esteemed guest speaker, Dr. Abhijith Kuttanamkuzhi, for accepting our invitation and gracing us with his invaluable insight. Dr. Abhijith expertise in the knowledge in the field of cancer biology, particularly in breast cancer research, are truly commendable. As a postdoctoral scientist at the renowned Princess Margaret Cancer Center in Toronto, Canada, Dr. Abhijith has been actively contributed to the advancement in our understanding of cancer and its treatment. Dr. Abhijith's insightful presentation has shed light on how research are explored the use of epigenetic targeted drugs to overcome resistance to standard breast cancer treatments. The area of study holds great promises enhancing treatment outcomes and improving the quality of life for breast cancer patients. We are truly honored to have Dr. Abhijith share his expertise with us, and I believe that his contributions will leave a lasting part on our research community. The knowledge gained from today's webinar will undoubtedly inspire the deep of this continue making significant step in the field of cancer biology and patient care. Once again, I thank to Dr. Abhijith Kuttanamkuzhi for being a part of this informative event, for sharing his invaluable thought with us. Ladies and gentlemen, so fasten your seatbelt as we embark on this extra exhilarating journey into this world of in vitro clinical models in breast cancer research, important of patient-derived organoids. Before we proceed to share to the slide with our guest speaker, so I am addressing the significant highlight to the top of this webinar. So breast cancer is a significant global health problem concern. So, and finding novel methods for early detection, prevention, and improved therapeutic approaches in crucial positively impact patient outcomes. In the past, in vivo mouse models were extensively used for preclinical studies and drug development research. However, transferability of results and animal models to human has been a major challenges, leading to the high failure rate of drug during the clinical trials. The difference in cellular and molecular processes, species and patient to patients, heterogeneity play a significant role in the descriptions. So, the recent advances in the clinical research have led to the development of patient-derived organoids and xenografts models, which better receives the in vitro scenario in humans. These models have shown that great potential in drug developments, target identification, and validation studies. Notably, breast epithelial-based organoid and cancer-based spheroids models have been widely utilized in these purposes. So, extensively recent update from the Government of India and LDS were promoted to the utilization of alternative model systems such as organoid and organ-based chips have been removed the requirement for animal-based testing in drug trials. This significant a positive steps towards more ethical and relevant research models. So patient-derived organoids not only offer the easier and faster testing platforms, but also have a potential to contribute significantly to the development of personalized therapeutics and drugs. As a result, patient-derived organoids are forced to become the present and future drug development, supporting faster and better advancement in the critical area of research. So today's webinar promises to the unique and enlightening as we explore the potential of patient-derived organoids in breast cancer research. By understanding the harshness, the power of these innovative model systems, we can make significant strides in the fight against the breast cancer and improve the life of patients worldwide. In this overview of this webinar, I would like to insightful and important of the webinar in this critical field. Our guest of speaker, Dr. Abhijith, will be brief about the experimental research. Now, Now, audience, before we begin our webinar and introductions our esteemed guest, allow me to provide a brief overview of Heredity Biosciences. Today, mark a momentous occasion as we present the remarkable journey of Heredity Biosciences, a provider of research and training services in the field of life sciences and healthcare. It is established in 2018 and reconstructed in 2021. Our company has earned trust from doctors and research and commenting our positions as a reliable and innovative partners. Backed by a team of experienced professionals in the life sciences, healthcare, and analytical and metabolomic services, Heredity Biosciences is dedicated to delivering exceptional solutions and cutting-edge products. Today, we are thrilled to announce the inaugurated in our new Guwahati, a significant milestones that expand our reach of impact. At Heredity Biosciences, our unwavering commitment to scientific innovations drives our processes. Our team, dedicated researchers and professionals, continuously explore novel experimental methods to better invaluable insight into various life sciences and healthcare related aspects. Our diverse portfolio of services like genomics, proteomics, metabolomic services covering a wide to the life sciences domains. We extend our services across India, ensuring accessibility and convenience for client and national boards. Our crucial area of focus on this clinical microbiology, infection genetics, cancer genetics, fertility genetics, conventional tissue-based DNA studies. Those services which strive to contribute to understanding, prevention, and treatment to various disease conditions. So we extend our gratitude to all these client partners and the team members who have been integrated part of this journey as we contribute to the commitment to pursuing the boundaries of scientific exploration and making a positive impact in healthcare and research. Now, Heredity Bio Academy is dedicated to excellence in life sciences training and research. We are proud to be leading training institute in Odisha. Our comprehensive hands-on experimental research training cater to the student and professionals, providing the invaluable hands-on skills knowledge with a diverse range of training modules, including clinical microbiology and genetics, clinical microbiology, nanotechnology genetics, food microbiology and genetics, and many more. The participants receive a well-rounded education in these chosen fields. We are thrilled to announce for the specialized modules such as clinical microbiology and genetics, histochemistry, and immunohistochemistry, which address to the latest advancements in the clinical field, ensuring the participants stay back to the science. Our next generations of research and professional in it is our milestone. Our training programs offer a platform to individuals to excel in their career, contribute in the life sciences and healthcare. We extend to heartfelt appreciation for all the supporter who ready to support and others introduced to the past participants. So we are learning, there are more learning experiences. Our team experienced trainer offer invaluable guidance and mentorship, including a rewarding journey, learning journey to the students. The genomic and microbiology lab is well equipped to support various research initiatives and enabling practical learning, hands-on learning to encourage participants to engage to the experiment, fostering the practical skills and independent learning knowledge also, hands-on skills. It is listen assistance, promoting knowledge demonstrations, collaboration with the scientific community, and placement systems. So, all interested in at Heredity Biosciences, the past 5 months have been filled with a remarkable advancement and a fulfill of immense pride. We have provided valuable opportunity more than 600 students, 100 training program, session of the Bhubaneswar Guwahati side. Additionally, more than 300 students completed in the internship program, gained experience in various research areas. Our students have achieved outstanding success in placement activities, ensuring position. Meanwhile, our dedicated teams has contributed significantly to the scientific knowledge, publishing more than 50 research papers in biosciences. We have fostering strong collaboration with esteemed hospitals like SCB, Kings, some hospital and other research institutes, facilitating impactful connection between the academia and healthcare. This collaboration is situated in experimental research to addressing critical healthcare challenges to improve the patient's care. With this advancement, Heredity Biosciences remain committed to advanced biosciences and improving the next generation of research. Now, ladies and gentlemen, it is my distinct pleasure to introduce our esteemed guest speaker to today's webinar, Dr. Abhijith Kuttanamkuzhi. Dr. Abhijith currently serves as a postdoctoral fellow at the renowned Princess Margaret Cancer in Toronto, in Canada. Dr. Abhijith's academic journey has been marked by excellence and dedications. He obtained his doctorate of philosophy in oncology and cancer biology from the prestigious Indian Institute of Science Education and Research, Pune. Prior to that, he pursues both in master and bachelor degree in biotechnology from Indian School of Biotechnology. Throughout this academic career, Dr. Abhijith has gained valuable research experience in various esteemed institutes. Notably, he served as a summer research fellow under the guidance of Dr. Tejani Haldar in the Laboratory of Chromatin Biology and Epigenetics, founded by the Indian Academy of Science. He has contributed as a summer intern in the Center of DNA Fingerprinting and Diagnosis and undertook research internship at the Indian Institute of Technology, Bombay, under Dr. Santanu Kejos. His expertise span a wide range of areas, including preclinical studies, stem cell research, biology, cell and gene therapies, and many more. Dr. Abhijith is recognized for his contribution, receiving several honors and awards, such as IITB, IRCC Research Fellowship, Internship Awards, Best Oral Presentation Award, Phenotypic Heterogeneity as a Drive of Cancer Projection Conference, Best Poster Award, Presentation Award.
Excuse me, sir. Your screen is not visible.
Now it's visible.
No, sir. You have to share.
Is it visible?
Yes, sir. Yes, sir. Yes, sir.
So, thank you. His expertise span a wide range of areas, including preclinical studies, stem cell research, biology, cell and gene therapy, and many more. Dr. Abhijith is recognized for his contribution, receiving several honors and awards, such as IRCC Research Fellowship Award, Best Oral Presentation Award at the Phenotypic Heterogeneity at a Tribe of Cancer Positions Conference, Best Poster Awards at the first PCCA conference and workshop at the 14th Indo Australian Biotechnology Conference, and the prestigious Newton Bhava PhD placements. With an immense list of publications showing his groundbreaking work, Dr. Abhijith's research has notably explored altered expression of antibiotic proteins API5 and its effect of breast thymogenesis, phospholipid-mediated induced transformations in the three-dimensional culture, prolonged exposure to platelet-derived factor, and its important on breast epithelial cells. A multi-factor researcher, Dr. Abhijith brings a wealth of in silico, in vivo, and in vivo expertise in this project. His skills in the preclinical model development, target identifications and validations, big data analysis, and the stem cell projection cell therapies underscores his commitment to advancing challenges in cancer research. As an enthusiastic cancer research fellow, Dr. Abhijith look forward at promising cancers in preclinical and clinical research, as well as pioneering work in cell and gene therapy and novel biologists. Let us extend a warm welcome to Abhijith as we eagerly anticipate an invaluable slide and he will share with our today's webinar in vitro preclinical models and breast cancer research, important of patient-derived organoids. Thank you for joining us, Dr. Abhijith, and we look forward to the enlightening the sessions ahead. This is our team, Heredity Biosciences, sponsor in Guwahati. This is our websites. Thank you.
Thank you very much, Dr. Purohit. Can I start sharing the screen?
Sure.
Thank you. First of all, thank you very much for this warm welcome and the introduction, Dr. Purohit, and also Ms. Sushri for coordinating this meeting, the discussion platform. I'm very happy to see know about the Heredity Biosciences and the different areas that they're working on. It's really proud to know that we have this kind of institution that are getting established and spreading knowledge about science in India. So, and thank you for all the people who are joining for the meeting. So I'll just start with what I was planning to discuss about. So as Dr. Purohit and Sushri had mentioned already, in today's meeting, I'll be discussing a little bit about in vitro preclinical models in breast cancer research and what is the importance of patient-derived organoids or PDOs. So before going into the PDOs or patient-derived organoids, we'll start with some basic understanding of what cancer and breast cancer is. So if you ask what is cancer, in a simple terms, it's a group of diseases that are characterized by uncontrolled cell growth. So as the name, as what it is said, it is where the cells of the same human body starts proliferating at an uncontrolled manner, and it loses all the control, or there is reduced apoptosis or reduced cell death, and that forms a mass of cells which then continues to proliferate and start to invade other part of the tissue. And what is known about initiation of the cancer is that it starts from maybe a single cell, when there is a genetic mutation in that, and it overrides all the mechanisms that are present in the organism that prevents uncontrolled proliferation or the regulatory signaling that is present within the cell and within the organism and start proliferating. They start forming something called as hyperplasia. So hyperplasia is a situation where there is like a mass of cells. Am I not audible? Is there anything that I should take care of?
Yes, sir, you are audible.
Okay.
Yes, sir, you are audible.
Okay. Yeah, so as I was telling, the hyperplasia once get established and it continues to proliferate and the cells continues to evolve out of that, forming things such as dysplasia, leading to in situ cancer, which is called as the benign tumors, and then finally leading to something known as invasive cancer. So once it is in the in situ cancer state, it starts, there will be more and more changes that are happening to different cells of this mass of cells. So that's being explained over on this figure, where there is a normal tissue and it starts having the first change. And this first change is followed by second, third, and maybe fourth and fifth changes, making a population of cells which are highly heterogeneous, having different varying degrees of changes that it has occurred. And all these together make up something called as the invasive cancer. And that exactly says that how much heterogeneous these populations are and how much it would be difficult to understand these diseases. And because of this highly heterogeneous nature and being the disease that is formed because of these cells that are present within the human body, it is also one of the diseases that's extremely difficult to cure or target and to, uh, to reduce its potential. So because of that, if you look into the cancer statistics all around the world, there is a huge number of people who are affected by cancer. And breast cancer is a major cancer type in worldwide and in India, affecting women. And as you can see over here, this is a statistics from 2022. Around 24% of all the cancer patients in the world are affected at the breast within females. And what is more important to look at is the incidence to mortality ratio. So this is a graph of about India, where if you look at the incidence to mortality ratio, it's almost 1:2. As when we compare that to Western countries, where the ratio is very, very, very low. And this actually says that it's a very important thing to now finally understand the need of novel therapeutic targets, identifying better treatment options, and also to find out mechanisms that can be used for early detection of cancers, and especially in countries such as India. Now, what makes breast cancer so important, or why is it it's a very distinct cancer? For the understanding that, we first have to understand how the breast morphology is. So if you look into breast, it's an organ that undergoes tremendous number of cyclical changes during the lifetime of a human being, of a female. And this is basically, the breast consists of glandular structures that secretes the milk, and these are called as acini. So as you can see over here, this is a cross section of the breast from a human mammary gland, a normal human mammary gland, not a cancerous tissue. And as you can see over here, they form these structures called acini. So the acini is a bilayered structure. In a bilayered structure, there is a myoepithelial lining, which you also call it as basal cells, and there is a layer of cells called as luminal cells. And these luminal cells are the cells that secrete milk, and this milk is secreted to the lumen, and this milk is then being carried through something called as the ducts. So if you look into the morphology of breast, there you can see that these are the things called as lobules. So within the lobules, these acini structures are there. So these you have to think these acini structures as spheroids. So there are a lot of spheroids that combine together to form something called as the lobules. And these lobules are connected together through ducts. And these ducts takes the milk towards the nipple. So the entire the organ undergoes numerous number of cycles of proliferation, apoptosis, following the hormonal signaling throughout the lifetime of the female. And what happens during this is a very phenomenal event where if you look into the normal menstrual cycle, within that there is a numerous number of changes that happens to this particular tissue. So if you start with the early follicular phase, when there is low estrogen and progesterone, and you consider that as a first starting phase, and when it goes to late follicular phase, there will be a spike in the estrogen levels. And because of the spike in the estrogen levels, what happens is there is an increased ductal expansion and fat deposition of the breast. So what do you mean by ductal expansion is that if you remember from the previous figure, these ducts that are present, it starts to spread around. So the lobules are yet to be proliferated and spread out, but then the duct starts spreading out. And then in the next phase, which is the luteal phase, you see a spike in the progesterone, and there is a decrease in the estrogen first and then a small increase later. And this is when there is ductal proliferation that happens, and it starts to extend to other regions and starts proliferating. And if there is no pregnancy, all these changes that have happened would return back in a process called as involution during the late luteal phase. And all these cells has to undergo apoptosis. So this cycle continues at every menstrual cycle. And then when there is pregnancy, things get more and more different, where upon pregnancy, there will be an enhanced proliferation, differentiation, and vasculogenesis that happens within the breast. So as you can see over here, this was how it started when a female is in its embryo state. And once it's in the puberty, you have these things called as terminal end buds that exist within the breast. And this starts spreading and spreading and going back during the menstrual cycles. But once there is a pregnancy, it starts proliferating and differentiating into all those structures, all these acini structures, and all the lobules that are to be spread all around the breast. And when the lactation has to happen, all these would be at the maximally proliferated, maximally enhanced phase where it starts secreting milk. And once all the involution starts, again, all these things has to go back. All those cells has to undergo apoptosis, and the progenitor population has to be regenerated. And this is a very important cyclical changes that happens. And because of these cyclical changes happening, it is also important that these cells should be having a very tight regulation of the signaling that is present within them. Because this continuous number of proliferation and apoptosis can also trigger genetic modifications because of DNA damages that happens in that can be like just because of the normal proliferative stress or the replication stress that this cells undergoes, or it can also be because of the types of drugs or the radiations that happens during the lifetime. And what happens in such scenario is that it can lead to scenarios such as breast cancer. And what you see over here is a cross-section of a breast cancer tissue. So breast cancer is, as I said earlier, it's one of the most common cancer in the woman. And it's very often associated with age, like it's most often diagnosed at an age about 50. And it can be majorly classified into two. There can be familial cancers and there can be sporadic cancers. So as the name says, familial cancers are something that is because of a genetic mutation that is running in the family. And the sporadic cases are something that is not associated with a heredity. And then this can be further classified based on various factors, such as the hormone presence of hormone receptors, or the type of the cells, the stage, the grade of the cancer, etc. Now, if you look into this is a cross section of the breast cancer tissue. And if you remember from the previous figure how a normal gland was looking and how now you can see how a cancer tissue would be looking. You don't see any structures. You basically don't have any duct. There are no lobules. You just see a mass of cells. And these tumors that are present can be either in the lobular regions or it can be in the ductal region. And based on that, you call it as ductal cancers or lobular cancers. Now, it is still a matter of debate to understand whether all the ductal cancers originate from the duct or the lobular cancers originate from the lobules. Well, there are so many theories that are existing. And finally, what you see is something like this. When it reaches to an invasive stage, you would be seeing just a mass of cells. And it is extremely difficult to identify exactly what are the drugs that need to be used, and targeting them is more difficult than that. The major problem with breast cancers is also that it is very, most of these cancers happens in regions that are pretty close to lymph nodes and where there is vascular genesis have happened. And this also leads to metastasis to other organs in the human body. And making this one of the major cancers that needs to have better treatment options coming up. And we also need to understand this cancer in a much more detailed way than that we know as of now. So talking about drug development, if you look into that, it's a very, very intense story where you need to spend like years and years of research and trial and errors to get that one compound, which would be the most ideal option for treating the particular cancer or any disease. So if you see, this is like a long pipeline of process that goes from thousands and thousands of samples to that one specific chemical compound or an antibody that nowadays have been used. And this starts from something called as stage one, which is the drug discovery. So this first stage is where you have to find what is a target that you want to target, and then identify what are the hits that you can use to hit this target. And this is where most of the time that the early preclinical or the first drug discovery time goes off, because you would have hundreds and thousands of research that is happening all around the world, and they come up with different ideas and they come up with various different molecules that can be utilized. But then you need to find all those which would have an immense potential, and then get a get a list of all these things and figure out which all can be used. And then you figure out what would be the best assay to screen all these compounds. And then to do a high throughput screening and get a number of leads that are there. Now, after getting this, the lead has to be optimized, and then you go to the preclinical studies. So preclinical studies, which we'll be talking more about today, it basically involves a lot of understanding of how the drug would be working, how exactly would it be functioning. And this involves utilizing various model systems, which we'll be talking in a while, and then also to understand what would be the formulation and how optimize what should be the dosage, that dose ranges that you can use. Once you have all those ideas, you go for a new drug application, and then it leads to clinical development. And the clinical phase contains phase 1, phase 2, phase 3, where phase 1 is a study on healthy volunteers, phase 2 and phase 3 are on patient populations. And this is where, again, you will need to have modifications based on the patient, and you need to understand what's the safety, what is the efficacy, what is the pharmacokinetics, and all those things. And then finally, if it all passes through all these things, it reaches for the FDA review and approval. And even after a drug comes into the market, the FDA does continue to do a post-market monitoring to confirm that there is no long-term after effects or side effects that are happening. And this takes a huge number of years. And if you see an approximate calculation of this, it's almost 3 to 5 years that is spent on this area. And the rest of the things are basically on patients, and it would definitely take a long time. And it's hard to reduce this time if you want to have a very effective and a safe drug. So things that can be enhanced or things that can be made faster is happening here. And that is the stage 1 and stage 2. But the stage 1 drug discovery, now with the advancements in the understanding of bioinformatics and the various AI tools that currently we have, there's an immense potential of utilizing them to do a lot of this in a very fast manner. But when it comes to the stage 2 preclinical studies, we still have a lot of hurdles that we need to come up. So the preclinical studies, as I mentioned earlier, it basically is performed in an either in vitro, in an in vivo, or in an ex vivo, or even in silico models. And this gives you an idea about exactly what you're targeting and what are the different, how is the mechanism of action, what is the biochemistry of these things that is happening there, what's the molecular biological understanding of how the downstream things are happening to this treatment. Now, this is mainly helping to understand the efficacy, like how effective these things would be, and then what is the mechanism of action. And then you also need to do certain quality control tests to exactly understand what would be the potent state and what would be the consistency, can you do, what is the storage conditions that you would need for this drug and all those things before you go for filing a new drug application. And these things, as I mentioned, are mainly done using things such as this in vitro and in vivo methods. And traditionally, we have two major techniques that is being utilized. First one is 2D cell culture. I would like to expand this a little more and say that these are cell line based studies. So a cell line is basically an immortalized cell that can be derived from a specific type of cancer. It can be derived from a specific tissue. It can be, there are normal cell lines, so-called normal cell lines. So again, the normal cell lines are still not normal because they are immortalized, means they have an immense potential of proliferation. They just continue to divide forever, while a normal cell in an organism does not divide like that. They have a finite lifespan. These cells has to undergo a process of cell death at a particular phase of this lifetime. And these 2D cell cultures or 3D cell cultures or spheroid cultures that are based on cell lines have been used for tremendously for various preclinical development studies, because it's a very simple, standardized, cheap, and easy to work around method. You already have cells that are in culture. People have already developed all those cell lines being there, such as, for example, for breast cancer, we have cell lines such as MDA-MB231, MCF7s, and etc. And these things can actually define specific types of cancers. And these specific type of cancers can be, you can easily use these cell lines and say that, okay, you treat with this particular drug and you can target this specific type of cancer. And it is also very easily modifiable, like you can genetically incorporate mutations, you can remove or knockout genes that could mimic what could be the scenario of the patient. However, the major disadvantages of this is that these are homogeneous populations. There is no factors of this thing, entire thing is not taking into consideration of what the exact situation is there inside a cancer. As I said in the initial slides, the entire tumor is a very heterogeneous population. You have different types of cells that are undergone different degrees of mutations and changes and different various levels of biochemical and molecular biological changes that have happened to it being there. And it's extremely difficult to explain all these just with one standard cell line. And most of the things are in static conditions. None of them are in any it doesn't give you any physiological relevance, and it cannot give you any clinical actual clinical predictions with that. So that's where the mice models comes into picture. The mice models, so what is being traditionally done is that you do certain number of assays using the 2D cell cultures or the cell line based studies, and then you go to the mouse models where you can use xenograft, where you can inject these cell lines into mouse, in an immunocompromised mouse, and then look into what happens to those tumors that are found because of that. Or you create a genetically modified mouse that can start forming tumors at a specific time of its lifetime, and then you try to treat it with specific drugs. Again, it's very nice because it gives you a lot of understanding about the whole body pharmacokinetics and the side effects that it have. And it also can have an understanding about how the tumor microenvironment would be. And again, it's easily genetically modifiable. Mouse models have been used for years and years for this kind of research all around the world. However, they also have certain disadvantages. First of all, if you use immunodeficient mice, that actually takes away a major factor that is involved in the tumor formation or the carcinogenesis. And these things, the mouse model, even though we have tried to mimic a lot of things with it, the genetic makeup and all those things, it is still a murine microenvironment. It is still not as close to what is there in the human microenvironment. And sometimes this takes a very, very long time to do all these analysis, taking up a lot of time in the preclinical studies. And for example, certain mouse models are there for breast cancers where you have to wait for 8 to 10 months to start seeing some tumor formation. And that is a very long time. You are almost losing a year, and maybe it would be successful or unsuccessful. And if it becomes unsuccessful, you have to start all the way again and then go back and look into some other options. And that is where the alternative models comes into picture. So these are advanced strategies where you can actually make clinically relevant, which are more close to the human in vivo scenarios, systems such as the PDXs and the PDOs. So PDXs are the patient-derived xenografts. And as the name says, both patient-derived organoids and patient-derived xenograft takes the patient tissue and puts it either in the mouse, calling it as PDX, or you culture them as in vitro to form these things called as organoids. Before we go to PDOs, we'll have a brief understanding about PDXs. So patient-derived xenograft that uses patient-derived tissue samples, and it's been passaged again in an immunocompromised mouse. And how this happens is, for example, a cancer tissue is being taken out either during diagnosis, where a biopsy is done, or during a surgery where the doctor actually removed the tumor, and this tumor tissue can be processed and made into small fragments and injected into these immunocompromised mouse. And these immunocompromised mouse can then be incubated to let them form the tumors. And once they establish these tumors, this can be passaged over generations. And you can have a PDX biobank of these mouses that bear these PDXs. and you can do whatever drug treatment whenever you want to, and get understanding about how these drug efficacies and how would be the how effective would be the drug and how safe would be the drug in a mouse that is bearing a human tumor on it. Now, it is a very nice system, and there's already been a lot of models that is already established for breast, colorectal, lung cancers, brain cancers, etc. However, this still takes a little more time because you need to wait till this human tissue gets incorporated into the mouse, and then you also have to understand that the microenvironment, even though it exists, it's a murine microenvironment. You are putting a human tissue into a murine microenvironment, and that actually does not give you the exact scenario that is happening within the humans. The immune cells, if it is not an immunocompromised mouse, or if the other extracellular matrix factors, the extracellular components that are there, and the cells that are present in the tumor microenvironment are pretty different between what is there in the humans to what is there in mouse models. And that is why it is important to make a model that can be more closer or that is more relevant to human scenario. And that's where the PDOs come into picture. So patient-derived organoids are basically three-dimensional cell cultures. It's just derived from the patient tumor tissue. And this PDOs actually retains all the genetic and the molecular characteristics of the original tumor. Now, you might have heard about things called as spheroids, and now we are talking about organoids. So it's not the same thing as a spheroid and an organoid. Spheroids are basically derived from cell line monocultures, where you have that cell lines that we were talking about earlier being cultured as three-dimensional cultures, and they form just a mass of cells. And this mass of cells actually does not form any resemblance to the actual tumors because it's based on that one particular cell line that is there. It won't be able to recapitulate exactly how the heterogeneity that is there in the patient tumors. And they does not represent the different multiple lineages that are present, while the organoids can form multiple cell lineages growing together in a particular morphology and characteristics that maintain what is there in the in vivo scenario. As you can see on this figure, now these are the normal breast-derived organoids set up by a group by Rosenbluth and his group. And what it shows is that what they did was they had normal breast tissue, which was then tried to culture as organoids. And they supplemented the required growth constituents, the growth factors, and the required media. And they formed these beautiful structures that have the same bilayered acini. And cytokeratin 8, which is green color, CK8, what is written here, is a marker for luminal cells, and CK14 is a marker for the basal cells. And you see that two layers of these cells. And this is exactly what is how in the actual in vivo breast acini. And this very nicely is able to be recapitulated in an in vitro scenario. And what's more relevant, or what makes it more beautiful, is that when it comes to cancers. So this is a study done by Bhatia et al. And what you see here is on the left side are immunohistochemical analysis being done between the patient tumor and the PDOs derived from that. If you see very closely, the structures of the cells that are in the tumor and the TNBC PDOs are pretty same. So they were able to recapitulate the exact morphology, the same characteristics of how the patient tumor was. Same with another sample. And this is already been established for a lot of breast cancer patient samples all around the world. There is already several biobanks that exist around the globe that creates these a bank of organoids. Like we have banks for cell lines, we have banks for organoids, where it will be a more standardized way where you can, when you have a drug that has to be tested, you can contact these biobanks and get the particular samples that probably are relevant for your study, the subtypes that were relevant for your study, and then do the analysis on them. Now, again, we might still have a confusion between why, again, this is very important, our PDXs, because we still haven't said about the other factors that are involved. So still, there can be a lot of differences between the PDXs and PDOs. So if you look into that, both the things are derived from patients. However, PDXs have advantages such as the tumor microinteractions are still preserved. And this has all genetic stability and the clinical responses can be predicted and all those. While, as I mentioned earlier, we have certain shortcomings in that as well. And using animals in itself is a issue that we should take care of. Like recent understandings have also, recent studies have also accepted the fact that we need to reduce the usage of animals. And the advantages of the organoids, it almost takes into the picture of all the advantages that the PTXs have, except the tumor microenvironment interactions. And then it provides much more than that. Because the PDOs are easily culturable. It is not going to take like 9 months to get it into culture. You can get them into culture as early as 2 to 3 weeks. And then you can start having all the analysis being done on this very early, right from the patient tissue has been dissected out. And it might take like 2 months to have the first set of results coming out. While we still have certain challenges that need to be taken care of, you need to understand that we will go into those things towards the end of this talk. So we currently have an idea about why the PDOs are a really good option. But then how difficult it is to set up these PDOs and what are the steps involved in setting up a patient-derived organoid? If you look into that, it's a very simple process. Like when a patient undergoes a surgery or there is a biopsy sample coming out, you get that sample, make sure that it is not getting dried up or it is the cells are not undergoing death, it's kept in a temperature controlled manner, and then you fragment it and you make it into small pieces and then start culturing them as organoids. And what you see on the right are the organoids that are formed from either normal breast tissue or from the breast cancer tissues. To have a further understanding, if you see, this is a breast cancer tissue that has been taken out during surgery. And a very small fragment of this is taken for setting up the organoids. You don't need the entire chunk. You can still preserve most of this. And this entire thing would help us set up and do research and do drug discoveries for several decades of clinical development studies. And these small pieces of tissues are then taken and cut into small pieces, digested, and then you remove all the red blood cells or cells that you are not interested in, and then you take them and put it in something called as extracellular matrix. So the tissue is dissociated, and then these are mixed with something called as the Metrigel or a BME or a hydrogels. These are basically extracellular matrices that are derived or synthesized based on the understandings of the tumor microenvironment. And then you prepare the media that is required to be supplemented. This would have the growth factors and all the other growth constituents. And this can vary very largely between tissue to tissue. But what is beautiful with breast cancer or the breast tissues is that we already have a very vast understanding about how we can set up breast cancer organoids or the breast cancer-derived patient-derived organoids. And then you plate them as either DOM structures, like a drop of the structures with the cells or the tissues inside it, or you make a bed of this extracellular matrix and then you can culture them on top. These are then incubated in a temperature-controlled, oxygen-controlled, CO2-controlled manner. And as early as 10 to 14 days, you will start seeing structures. And those structures, what do you do after having those structures? So once we have structures like this, what can be done is a numerous number of analysis methods. And this numerous number of analysis methods can actually answer most of the biological questions that would be relevant for a clinical study. And for some examples that I can show you here, this is an example of a flow cytometry. So you can look into things such as protein expression, how the cell cycle profile is, how fast the cells are proliferating. And what happens if you treat with a drug? Does that stop proliferating? Does that have a different expression of the proteins? You can take the cells out of these organoids and do this analysis. You can even take the cells out of this and select a particular population that you're interested in, put it back and see what happens to it. You can specifically select things that are involved in a specific lineage of the cells that is there, and then take them out, culture them separately as organoids, and then see if you can target those with a specific drug. Also, omics have become like a very majorly used methodology all around the world. What you mean by omics is understanding the mRNA expression, the protein expression, which includes the proteomics, transcriptomics, and metabolomics. And you can also collect lysates or take out exact proteins or RNA or DNA from this and carry out things such as proteomics or metabolomics or transcriptomics, as well as genomics, where you look into the different mutations, if the tissue have a particular mutation. And all these are done from a small amount of tissue that comes from the patient. You don't need to get a huge chunk of the tissue, or you don't need to have the patient coming again and again and again, or you are not treating the patient again with these drugs to see if there is an effect. And more than that, with regards to preclinical studies, you are not actually utilizing any cell lines that were established for decades ago from one single patient or one single human being. And you can actually get all the various heterogeneity within the population and see what exactly happens when you treat a particular drug on these breast cancer organoids. And what's more beautiful would be to get these organoid sections and do all the histological analysis over here. You can, which is not even remotely possible with utilizing all the 2D cultures. You need to have 3D cultures for doing all these things, because then only you will get all these structures which will resemble what exactly would be there in an in vivo scenario. And finally, you can even look into the drug efficacies. You can treat these organoids and see how they are surviving. You can look at the different concentrations, you can look at the different time points, helping us to understand what would be the ideal dose range for treating these tissue cancers, or what would be the ideal time gaps that you need to give. And for example, this is a study by Lee et al., which came in 2020, and they have looked at drugs such as tamoxifen and fulvestrant on organoid, breast cancer-derived organoids. And what they see, you're not only seeing what the cell viability is. This is what you can also get from a normal cell line-based cultures. But you can also look into what would happen in vivo in these structures. Are those structures going to die? What is happening within that? How these drugs actually affect these structures? You can see them. You can take these out and do all the sequencing, all the omics studies to understand what exactly is getting affected. And what would be the ideal drug dosage that would cause minimal effect on a normal cell that might be around and only affecting these tumor cells. So all these things can be carried out using patient-derived organoids. Now, another study by Gillian et al. In 2020 also showed that how close this can be to patient-derived xenografts. So patient-derived xenograft, as I mentioned earlier, it's considered important because it will help maintain all the tumor microenvironment interactions. And it still have a larger heterogeneous population. You are injecting it into an entire body of the organism. So if you compare what drug cells are being got from a PDOs versus PDX, they are very closely matched. This study shows that the drug screening shows concordance with the in vivo data. Whatever you are expecting whatever you were getting from the PDXs are exactly what you can get from PDOs with a short time. You don't have to wait for all these, and you're not using animals for the study. So this exactly says how important breast cancer patient-derived organoids are for drug screenings and understanding the preclinical aspects or different how the drug efficacies are, the molecular biology, and understanding how these drugs would be effective or functional. And you can also do modifications on this to make it more relevant and more beautiful. One of that is co-culturing. So co-culturing is where you got the tissue, and there are two ways of doing co-culturing. What you can do is you can just dissociate this tissue or make it into small fragments and do not kill any cells or do not sort for any specific type of cells, and then just culture them all together. You will have all the immune factors coming in. You will also have all the different other types of cells that are present all around. You will have the fibroblast, you will have the skin cells that are coming in, all coming into the same organoid structures. However, one problem that can be faced here is that it is difficult to have a media that can maintain all these things together. So an alternative method is to collect this tissue and maybe peripheral blood or other bodily fluids that would be associated with the tissue, the cancer tissue. And then you select and culture those cancer cells or the tumor, and then grow them as organoids. Once they establish as organoids, you take this fluid or the blood associated immune cells or things like that, and you can put them in at any concentration, at any whatever specific things that you would like to know about. And then this would make it more interesting and more closely relevant to the human in vivo scenario. And another very advanced methodology is organ-on-chip. It's a very booming field. People have been talking about organ-on-chip for a long time. Like recently, so many people have come up with methodologies that can be done. There are so many research that is happening everywhere, even around the developing countries and the developed countries. Altogether, they are trying to establish these systems. And what makes it very beautiful is that they have something like this, a microfluidics. They can mimic the cell stroma interactions, a nutrient gradient, or if there is some other gradient that is required, an oxygen gradient, a hypoxia gradient, how the metastasis would be happening, how the vascularization would be happening, what is the shear stress that is faced by these tissues. And you can basically mimic all these things. And this has been currently done very well with a lot of cell lines and single cell structures. Once we can get incorporated, there are studies already, like this is a 2021 paper that shows about how a tumor biopsy sample was being kept in a microfluidic and how it could relevantly recapture everything that happens here. And this covers all the things that we were missing from the PDX. And this makes it such a beautiful system where you have everything that can be done using a patient relevant tissue sample and in a very fast time. You don't have to wait for months to do all these things. And the other part of the story is these PDOs are not only going to help the preclinical development. These are also very important for personalized medicines. So what I'm going to show you about are two companies that work on something like this. Crown Bio is a company that works on utilizing these patient-derived organoids for doing personalized medicine and figuring out what would be the ideal therapy for a specific cancer. So as we have been talking about, the tumor is highly heterogeneous, and it is heterogeneous within the tissue, within one particular patient. And more than that, it is also very heterogeneous between different patients. And that makes it extremely difficult to understand and use a single therapy for all of them. So that's why people had started to classify them as subtypes, so that you have a particular subtype and you figure out, okay, this patient is this particular subtype, you can give this particular drug. And personalized medicines makes it to the next level. And PDOs would be the best way to do all these things. Because once a biopsy is done from a patient, for example, a breast cancer biopsy is done, and then these are cultured as organoids. And Crown Bio actually carried out a lot of omics screening to figure out whether there is mutations, what are the different genetic makeup of the different population that is there, and then also do a drug screening to figure out how these organoids would be responding to the specific drug treatments. Now then, have an understanding about the entire thing and go back to the patient within few months and then say that, okay, this is probably the best way of treating your cancer. And Forecast Biosciences, which is another company based in Bangalore, they also do something similar. They take out the tumor, they also take the peripheral bloods, and then they culture them as fragments or organoids, and then they have established methods to do the flow cytometry and all the analysis that we were talking about. So the PDOs are not only going to be relevant for just preclinical development, it is going to be relevant for a large number of things that are present all around for cancer therapies. And breast cancer being one of the major cancers affecting different countries all around the world, and also one of the cancer that numerous number of research have already happened. A very nice number of studies have shown different subtypings and a great understanding about what are the possibility of targeting this. And every day we get new researchers papers coming out saying about possible new drug targets. And patient-derived organoids on these breast cancer samples have pretty nicely established where we already know what are the components that are required, what are the growth factors that are required, what would be the growth constituents that are required, and how all we can modify this. And currently, we also have all the systems where we can analyze these samples. And we have infrastructures all around the world where you can get these things done in a pretty fast manner. And this is also supplemented by government agencies and the agencies that finally approves the drug, such as FDA. FDA in January came up with a new update where they no longer need to require animal tests to be done before human drug trials. And what they said is that it will help end the needless suffering and death of animal test subjects. Yes, so these animals undergoes a huge number of stress, and when these have been injected with different types of cancer tissues, they are literally going to bear all the pain. And then finally, when all this happens and then the drug fails, it's a complete waste. And this also will get us safer, more effective drugs to market more quickly by cutting red tape that is not supported by current science. And this is very true. And followed by that, India also takes the same step. It removes animals from drug testing process. A new rule authorizes researchers to use non-animal methods to test new drugs. The amendment authorizes researchers to use non-animal and human-relevant methods, including technologies like 3D organoids, organ on a chip, and advanced computational methods to test the safety and efficacy of the drugs. And this is very interesting as we have been now talking about all the PDOs. We have been understanding how these patient-derived organoids are very relevant and how we can recapitulate everything. But it will completely become irrelevant if the government agencies and if the pharma companies are not able to adapt it. But it's a very happy beginning to see that they understand these facts and they also are in the same line and accepts results from this. But nevertheless, we still have certain challenges. I'm not saying we have a perfect situation here. The challenges involve, some of the challenges that we still have with this is the PDO models are limited to certain cancers. Like we have been talking about breast cancer derived organoids now. They have very beautifully established organoids that can recapitulate everything that is there in the tumor tissue. But it is not there for all the tumor types, all the organs that are affected by tumor. So we need further study to understand this. And current studies, it still lacks certain cell-cell interactions, cell microenvironment interactions, cell extracellular metric interactions. And that is where we need to start combining the organoids into organ on a chip. And none of these methodologies, in vitro methodologies, will work as a single one. You need to combine all these things to get one of the best models that can be used for drug screenings. And now consider this for preclinical studies and for personalized medicines. For preclinical studies, it's a much cheaper alternative for all the mouse-related studies as compared to the mouse-related studies. However, the analysis that are related to it, if you want to do certain things, it is still on a slightly costlier side. And when it comes to personalized medicines, you still need to get the cost pretty down. Because if it is a personalized medicine, it's like every patient that goes into clinic and wants to understand or wants to figure out what would be the best drug that is possible, need to do the screening. And you need to keep it so minimum that it is affordable by every patient that is all around the world. And then what we have seen, what I have understood during the past years of when I was doing the work on these samples, is that some patients still fails to form these organoids. And these are small shortcomings which we need to understand why there's some certain number of samples. It's a very small percentage of samples, though we cannot leave anything unmet. And those understandings still needs to be figured out. And a very important thing, in India, we need to have an ecosystem, such as a biobank for these organoids, where any company, any pharma company that want to do preclinical studies on a specific drug can contact this biobank, and they have verified genetically identical PDOs from different cancers that are marked for different subtypes, and then you can do that. So this will, overcoming these challenges would make PDOs into a next level, and it can actually help us understand or facilitate faster, more relevant preclinical studies. To summarize, the mouse models have been successfully helping drug development. However, it has shortfalls. Majorly, first of all, it's using a murine system and all the problems that is associated with that, and utilizing animals for the cruelty and all those things associated with that. And at the same time, the lack of relevance to a human model system. And PDOs can resemble the in vivo scenario, and it can maintain all the structure, functions, and cell-cell interactions within that. Now, PDOs can also be faster. As I had mentioned, you can do a lot of other number of studies, and combining this with other methodologies such as organ-on-chip, it can actually complete the void and provide a very robust testing platform. And we are actually in the right phase currently. We are going in the right direction. So many researchers all around the world, pharma companies, research institutes within India, abroad, all around the world are working towards this. We have a very nice ecosystem that is being get up in several areas around the world. And it's the future is here. Thank you for joining. Open to any questions that you have.
Thank you, Dr. Abhijith.
Thank you. I see there was a few questions that are there on the messages.
Some of the question in the message box.
Yeah. Should I respond to that? Okay, I can answer that. Sorry, I didn't see that during the talk. Okay. So first question was about how BRCA1 and BRCA2 genes are helpful in identification of breast cancer, how these genes are identified, how mapping is validated. Okay, so BRCA1 and BRCA2 are associated with familial breast cancers, where the mutations in these have been shown to be, they go from, like as a hereditary, they go from the first generation to the next and next. And what happens is that BRCA1 and BRCA2 are associated with DNA damage response. And once the BRCA1 BRCA2 genes are mutated, the DNA damage response function is affected and the repair is not happening, leading to more and more mutations being present. And that actually takes away several that can actually cause the first change, as mentioned in the first introduction figure. And even a BRCA1, BRCA2 mutation carrier, they are not going to start having cancers from the first, because it's usually on only one allele of the chromosome. It need to undergo several other changes, and both the alleles may take the BRCA mutations, and they undergo they undergo a transformation. Those cells undergo a transformation, leading to cancer. Now, how these genes were identified? They were identified by understanding first how there was a family cancer recurrence that was happening, and then these were mapped to a specific mutations which included BRCA1 and BRCA2. I hope that clears the question. I can explain if you have anything more on that. And the next question was, are there any microchips available till now to study drugs of breast cancer for the preclinical trials instead of animals? So as far as I understand, there have been studies. There are a few things, several methodologies, organoid chip methods that are established. How often it is utilized is still a question, because even now these traditional methods of mouse models have been very widely used, and it's very recent. As I showed you, it's like this year we just had approvals from FDAs and the Indian government about accepting utilization of these models. So it will, I believe that this is going to be a change that we will start seeing pretty soon. And as you can see, there are already companies that are working in the field. So even not only the basic research, but the pharma companies are also accepting that there is possibilities with this. Okay. Manish had a question that how personalized medicines would be useful as everyone is having different genomes. It would cost great for developing countries like India. So what you can suggest for this? Exactly. So that's what I said. The challenges, one of the challenges that it's as of now, it's a difficult thing. You cannot do the entire genome sequencing for all the patient samples. So a major challenge in that is that for developing countries like India, you need to get the cost minimum. So you need to figure out methodologies where you can get a lower cost and do the analysis for that. However, maybe this is just my opinion, that a methodology for this is that first understanding the landscape of breast cancer in India. So basically all the clinical studies, most of the preclinical researches, all the organoid development mostly are currently happening in the Western countries. And what is different between the Western countries and the India is that there is a difference in the mutations that are very often found. So as the studies have done, BRCA1, BRCA2 mutations are very often found in familial breast cancers in Western countries. However, in India, there have been studies that have shown that it is not only BRCA1, BRCA2, there are other mutations that are also associated very commonly. And you need to understand those to have better targeted therapies. So you need to further understand the landscape of these breast cancer patients in India. And if you have a better idea about how classifying specifically the Indian breast cancer population, we might have an option to classify them and do some basic assays that will help understand, screen these and classify them into subtypes, and then give the treatment specifically for them. So you may not have to do the entire sequencing or genomic screening, but you can specifically do some of those and figure it out. Adintadi had asked saying, are these organoids already in use? Even in India, there are science research groups that are working in India, which already uses organoids. And as I said, there are a few companies that are also trying to establish this. And they have currently, I know a few companies that are Hyderabad based and Vizag based, which works on establishing these different tumor models and screening drugs. And then there are so many number of research groups that tries to incorporate these organoid-based assays into their work. And can these mimic in vivo conditions? Of course, yes, these can mimic in vivo conditions. And then are PDO and PDX two different methods? Yes. Or it is step-by-step process like first PDO and then PDX? Not really. PDO and PDXs can be two different methods and can be combined as well. You can establish a PDO and then take the organoids and inject it into mouse, and which is still a patient-derived xenograft. And while at the same time, you can also directly take the sample out of the patient and inject it into mouse. If these two methods are different, then which one would be best between the two? As of now, there is no best. You both of them are patient-derived methodologies. While PDOs, actually, the major importance of PDOs comes is when the ability to modify. You can make the required changes as much as you need, or you can modify the components, you can modify the factors involved, and you can do the changes in the genetics, and that is a little difficult thing with regards to PDX. And PDXs would take long time to establish and analyze, while PDOs are comparatively fast. What about AI models? Mohammad Faisal had asked, what about AI models like ChatGPT powered drug discovery, which can be used to find and screen compounds and test it for further research? Yes, so this is the part of in silico studies, the bioinformatics part. There have been a lot of development in this area. The bioinformatics have been booming all around the world, and in India, there's been so many groups that have been working on this. And AI just supplements this with so much power, because statistics on these informatics have shown very helpful, very nicely to identify novel drug targets and novel options for drugs. And this, although will not complement all the in vitro assays or all the patient-derived organoids or any preclinical studies, because the ChatGPT based AI models works on what is already known about all this, and they try to extrapolate that. Or any AI model would have a learning or the understanding about what is already known, and then tries to answer your question about that. So, which is a still evolving field. The breast cancer biology is still being as understood every day and day and out. We get new information about it. And the AI models would definitely help get streamlined initially. You can get do a certain amount of assays with that. You don't have to go and read about it, or get understanding about every single thing, but rather curate it in the AI and get a list of possibilities. But still, we will have to screen the possibilities and to actually see whether this can be used in humans. Will I need, Krishna Sri was asking, will I need any other test for breast cancer? Sorry, I exactly didn't understand what you mean by that. Like basically, it's a clinical doctor who would be looking, who would be first determining whether there is breast cancer or not. They are the final decision makers, and PDOs are going to help in identification of better treatment options for that. Before development of new drug, we have to know that the cells are resistant to the old drugs. So which method the drug resistant test performed on these cells? Drug resistant test have been very widely being done on the cell line based studies, but then obviously organoids are the better alternative for that. It's the same thing. So basically, for example, if you get a patient sample with a particular mutation and you want to see whether the drug that you are using is going to be resistant in this particular population, you can do the same drug assays where you get the organoids in culture and then you treat it with a specific drug at the drug concentrations that have been established, and see whether these cells or the organoids still survives at a time that where you see that those which respond dies. And that can actually show that there is a possibility that this specific drug can be, this particular tissue can be resistant to the drug that you are working on. Pritisha was asking, according to you, what is the most common cause of breast cancer, and mentioned few common symptoms for early detection. Common cause of breast cancer, there is nothing like a common cause. Most of the cancers that are sporadic cases. The familial cases are there, but most of the breast cancers are sporadic cases where you see varying degrees of mutations or gene expression profiles and all those things. And there are clinically, the doctors have identified several risk factors that can lead to breast cancer. And this also include hormonal associated things, and also obviously all the radiations and the different chemicals that you are you're prone to. Common symptoms for early detection, I am not the right person to answer that because that's a clinician who should be answering that. I won't be the best person to answer you for that. Okay, Abhishek was asking, is PDO can helpful to study further radiology effects? Yes, that is something that in my current work that I'm doing actually, where I'm trying to use these patient-derived organoids and you can even treat them with radiation and see how these PDOs would respond and what are the changes that are happening to the PDOs. How can we lower the chance of getting breast cancer? Again, that's mostly a clinician associated question. Yeah, I may not be answering for that. Another question is from Mohammed Faisal. So, in your slides, it was mentioned that breast cancer occurs in women after 50. I'm not saying that all the breast cancers happen after age of 50, but it's very commonly seen or associated with age. And that is not proved, though thought to be associated with the hormonal fluctuations, like the continuous hormonal up and down changes that are happening in the lifetime of a female. And the early detection, as I said, the methodologies currently we have are that is present, that is developed by clinicians. Although in several countries, we have methodologies including circulating tumors, identifying circulating tumor cells at a very early stage, or identifying the circulating tumor DNA. And these are things that are currently in developing, where you can do a blood test and identify whether there is a cancer being formed. And then there are these improvements that have happened to the scanning, where you can even identify smaller benign tumors at as early a stage. And one of the things that India still lacks is this early detection, where the clinic actually sees a patient when the tumor have actually grown to a stage 3 or maybe stage 4. So this also need to be part of the awareness among the population, that you need to understand and you need to screen for this if you have even slightest doubt. And also the major factor that affect India is the cost associated with this. So there should be options which are provided by the government agencies, which would have which will help screen the population and can identify at a very early stage. Okay, who will lead my overall treatment? Again, I didn't understand that part. What you exactly was there? Can we use this PDOs and PDXs for other diseases? Of course, yes. It is being also used in various other things, including PDOs are being used for even COVID-associated studies, because you can use them for how the lung would be infected because of COVID, and all those things were carried out even using PDOs. Biotechnology got any solutions for breast cancer by any means? I'm not a very pro in nanobiotechnology, but I think, yes, they do. They are specifically known for better drug deliveries because it helps in targeting the molecule that you are going to treat. Okay, if I'm worried about managing the costs of cancer care, who can help me? Who can help me understand that what aspects of my care are covered by my insurance? Sorry, this is not my domain at all. I won't be able to answer you for that. I'm really sorry for that. Are the PDOs potent enough to develop drugs to treat second and third stage of breast cancer? Yes. Can they help us with stopping the metastasis of other organs? So PDOs won't help you stop the metastasis, but what helps, the PDOs actually would help fasten the process of drug screening, both in a preclinical development and in a personalized medicine. So usually one major problem that has been faced by several clinicians is that they try to understand the cancer, they try to give them treatments, but then many of them end up having resistance, or that they would respond to the treatment, and they have to continuously change the methodologies. While when you're doing this, for a patient, when you're treating them with specific drugs, it can also affect the normal well-being of the human being. So those things can be at least slightly avoided by doing the PDO-based screening studies. PDO and PDXS is only for breast cancer? No, it's already, there have been so many different tissues where the PDOs and PDX are established, including brain tumors. What risks and side effects should we expect on a treatment lens? Okay, so if you're talking about the side effects of chemotherapy, it will greatly affect your normal well-being at many times. Although the clinicians have been taking care of this, they are trying to reduce the effect on the normal living conditions. They are trying to reduce the side effects. They maximally try to make sure that you have a better recovery time from with the treatments and all those. So these includes several things that a clinician would obviously be able to help you better, but I can just say that it would affect your digestion, your normal well-being, because you are treating your entire body with a specific drug. It's going to affect your other normal body parts also. So there is going to be some side effects, but that's where the clinicians comes into play, and they try to understand what's the best beneficial option for you, and they help you with that. Okay, as per American Cancer Society, Black women are most likely to suffer breast cancer other than any other racial or ethnic group. Is there any genetic or other reason behind this? There are various factors associated with this. It is not only like there are certain family groups, or there are certain populations where it has been identified there is a very high risk of breast cancer because of certain mutations present in them. While this can also be due to late detections, like in India, the scenario is there because of late detection and the cost associated with this. So these are things which can be easily or comparatively easy to identify and find a treatment option for that. First, what is the action of the governments towards it? I'm sorry, I didn't understand for what. Okay. Is there any way we can target the genes that might develop into cancer later, or by any bioinformatics technique to stop them even before they can transform? Unfortunately, it is difficult, but if it is a familial cancer, there have been genetic screens that are very commonly conducted all around the world now. So you can identify if you are in the risk of it. And with regard to options for preventing cancer development, we are still in a phase where we are trying to understand if there is a possibility for that at all. Are PDO and PDXS the only two solutions for breast cancer? So PDXS and PDOs are not a solution for breast cancer. It is a solution for increasing the feasibility, the time taken for developing drugs, and to find better options that are more human relevant. Dr. Purohit, if I should stop in between, you can tell me.
Yeah. One silly question from my side.
Yeah.
Breast cancer
I was
is
just
a
going through
genetic.
it. I'm sorry. Sorry?
Is the breast cancer is a genetic disease?
Breast cancer can be a genetic disease. Like as said, the BRCA1, BRCA2 mutations is a very well-known mutation that are associated with breast cancer. And there are other mutations that are very commonly associated with the DNA damage repair response signaling, being found very often in breast cancers. And obviously, it is very often as a familial cancer as well. So yes, it can be a genetic disease. However, there are other factors and hormonal factors especially affecting this. And just because having one allele of a mutated gene would not have cancer, there should be a number of changes that happens to the breast that where you have both the alleles having the particular mutations or other mutations that are the changes that lead to invasive cancer.
So what is the risk factor if there is a breast cancer is happen?
Sorry?
What are the risk factors if there is a breast cancer is happen?
So the risk factors, obviously, the family history is one importance. Like if you look into if there is the family have an history of having cancers, very common, you are in a higher risk state. And then obviously the other things that include are the kind of diet, because recently evidences have shown that metabolism also affects cancer development, because how the kind of diet that you're doing. And the other factors are obviously the radiations that are caused, or the chemicals that the different drugs, if you have other drugs that you're taking, those all can affect. And also, it is very often associated with the hormonal changes, the fluctuations that happen in a female, because the hormonal changes triggers a lot of proliferation and apoptosis in the breast.
Okay. Thank you.
Yeah.
Thank you, Dr. Abhijith. It's a
you,
nice
Praveen.
presentation and wonderful presentation.
Thank you so much.
So
Thank you so much.
everyone should be clear for this. Already a time is going on. So anyone who have a question, so you should drop a mail to Dr. Abhijith. So he should be directly messaged to him. I will drop to the mail ID to everyone. So everyone should be directly mailed to.
Thank you so much. Thank you so much.
Yeah, finally, this is a wonderful presentation. So on behalf of Heredity Biosciences, I extend my heartfelt gratitude to each and every one of you for being a part of a insightful, impactful webinar. So today webinar is a very, really truly is having a remarkable journey, breast cancer research. Thank you to our esteemed guest speaker.
Thank you so much. Yeah.
So your expertise and knowledge has been truly commendable, and it's my honor on behalf of this Heredity Biosciences and all these participants to hear. It's my honor to the opportunity to learn from you. Your presentation has been enlightened, offering valuable insight into the significance to the patient and derived organoids, and the role of this advancing breast cancer research. I also
thanks a lot. Thank you so much for the opportunity.
Yeah, our gratitude to all these participants who actively engaged to this webinar. Probably more 200 participants. This is first times actively engaged to this webinar. You have overwhelming response, and throughout the questionnaire sections, all this given the questions answers. Today's discussion made this event successful. I would really acknowledge to dedicated effort to my Heredity Biosciences team, Susri Devasis and Siram, and they are actively doing for this last 20 days. So thank you, Dr. Abhijith. So it's our honor, my honor also you are accepting our invitations. Once again, I thank you to sharing your expertise with us, and thank you all for the past participants, and to make this webinar a resound success. We look forward to the collaboration.
Thank you so much.
For the future, and such a nice event, and for the future we will for that. Okay, thank you, wonderful day.
Thank you. Thank you. Yeah. Thank you all.